to say that something is voluntary means that the central nervous system is in control of the muscle. The muscle is not really controlling itself like cardiac muscle or smooth muscle. This muscle is waiting for a signal from the central nervous system. What are the two parts of the central nervous system? What are the two main organs of the CNS? What are the two main organs of the central nervous system? Brain and spinal cord. Brain and spinal cord. So this could be coming either from the brain or the spinal cord. Brain or the spinal cord. Yep. And what we're going to see is what happens at this place between a neuron and the muscle, called the neuromuscular junction. So what we have in our book is a slightly different illustration of this neuromuscular junction. It looks a little bit like this. So this is the motor neuron, and this is the muscle. This is on page 112, and I'll get to it in just a second. But kind of going back and forth between some of the text on 111 and the picture at the bottom half of 112 is to sort of kind of talk through all of this stuff that are on this picture. This is the neuron here. I'll start labeling some stuff. I've taken a zoomed in view of just the part up at the top. Okay, let's see if I can label this. There's a neuron right there. Okay, this is the muscle here. Okay, so this is a phospholipid bilayer, brain right here. That's what that phospholipid layer around muscle is, the sarcolemma. That's what that represents. Okay, and so this whole arrangement between the neuron and the muscle is called the neuromuscular junction. Okay, so that neuromuscular junction consists of the end of the neuron, that's called the axon terminal, which is really just what's pictured here. This whole rounded end of the neuron, that's where it comes to an end. So that's the axon terminal, plus the motor end plate, which is this whole area. It's a part of the muscle. Motor end plate. That what's called a motor end plate is just this cup-shaped area, this whole part right down here on the muscle. Okay, so the axon terminal is part of the neuron. The motor end plate is part of the muscle. Okay, all right, plus the space between. So this whole thing called a neuromuscular junction is the axon terminal, part of the neuron, motor end plate, part of the muscle, plus the space between, which is called the synaptic cleft. Synaptic cleft. Whoops. That looks like deft. Synaptic cleft. C-L-E-F-T. Those three things together. Now this is over on page 110, bottom of page 110, kind of the summary of the whole thing. The axon terminal is going to release a chemical across the cleft that's going to bind to receptors on the motor end plate. So this all happens in that neuromuscular junction, that space where a neuron activates a muscle. Neuromuscular junction. Okay? So what happens at this neuromuscular junction. I'm going to erase all of this, kind of transition over to page 111. And again, you can be on page 112 labeling stuff too if you'd like. Just kind of know that I'm in the picture on page 112, but I'm sort of talking through the things that are described on page 111 so that if when you need to study this and you need to come back to it and look at it again, that's where I'm getting all this from. All right? Let's clear all this. And we're going to start over again. So what happens at this neuromuscular junction is an action potential travels down the neuron. Now, what an action potential is actually defined. So I'm kind of looking at point little letter A at the top of page 111. So page 111, point 4A, and it just says the action potential travels down the neuron. But down under point 4H, is it? Yeah, H is a little bit better definition of what an actual action potential is. An action potential is a switch of a change in the charge across the membrane. Now, in H, it's talking about a change in the charge across the sarcolemma. But what I want you to remember, I know this is probably going to sound really confusing at first, so I want to actually back up to talking about something that we talked about weeks ago called the sodium-potassium pumps. We talked about active transport that requires ATP. And sodium-potassium pumps pump sodium and potassium, as their name implies. But how do they pump sodium and potassium differently? They don't pump them the same. Remind me of how sodium-potassium pumps pump sodium and potassium. Anybody recall? Channels are different. Channels, tubes that are going to allow. We'll get to channels. These are pumps. These are pumps. So they require ATP. They need ATP, and they're called sodium-potassium pumps. They are called sodium-potassium. Yep, you're giving me the name. They're called sodium-potassium pumps. And what do they pump? Sodium and potassium. But they don't pump both of those ions the same. Okay. So I'm feeling like we missed an important point. Okay. So maybe this is new. Maybe this is new. They pump 3 sodiums out, 2 potassiums in. Okay? Does that ring any bells? Ringing any bells? Okay. Yeah. This is what, if it hasn't gotten confusing already, if it hasn't gotten confusing already, this is where it starts to get confusing. The pumps pump. The channels are more like drains. The channels are more like drain pipes. They are different. The problem is the channels are named for the ions that they let drain. So when you hear the phrase sodium channels and potassium channels, and then you hear sodium-potassium pumps, I think it's really easy to think that we're talking about the same thing, but I promise we're not talking about anything close to the same at all. So here's what I want you to notice about what I just drew up on the screen. 3 sodiums out, but only 2 potassiums in. And these things pump like 10,000 times a second, and there's thousands of them on the membrane. Thousands of them on the membrane. Okay? So what that basically means is for every 3 pluses that are pumped out, only 2 pluses get pumped in, and there's way more pluses outside the membrane than inside. And so the membrane is more positive outside than it is inside. And often we say that relative to each other, the inside is more negative. Okay? That's true of this neuron too. And to be honest, it's true of really every cell of your body. Every cell of your body, every cell of your body has sodium potassium pump. Every cell of your dog has sodium. Every cell of your goldfish's body, or maybe you have a betta fish, every cell of that tree outside your window has sodium potassium. Every cell of the bacteria on your teeth has sodium potassium pumps. Every living thing has sodium potassium pumps, and practically every living thing is a little more positive on the outside than it is on the inside. That's a grand sweeping idea that all life has in common. Okay? And there are very few concepts more important in understanding medicine and physiology and the function of all cells than the idea of sodium potassium pump. So I hope you don't mind me taking a few extra minutes to remind you of what the sodium potassium pumps do. And the sodium potassium pumps set up this arrangement of positive outside, negative inside, that we refer to as the resting membrane potential. Okay? RMP, or the rump for short, rump, RMP. This is how cells are at rest. Okay? That's a resting membrane potential. But what happens coming down this axon is known as an action potential. And an action potential causes the cells to temporarily be more positive inside and negative outside, a reversal of the charge. An action potential is when sodium comes flooding in to join the potassium. It comes in with the potassium, and now more positives are in than out. How does the sodium come in? How does it get in? It can't pass right through the membrane directly because it's got a charge to it, and it can't go through the phospholipid bilayer. It can't go by simple diffusion. It will go from high concentration of sodium to low concentration. Yeah, Krista got it. It needs a channel. And what do we call a channel that lets sodium pass through? This one's too easy. What do we call a channel that lets sodium pass? Yeah, a sodium channel. Yes, a sodium channel. But that's, like I said, that's where the name gets confusing. A sodium channel is going to let sodium diffuse. So, Heba, you were correct that diffusion would be the method that it gets into the cell, but it has to pass through a channel that will open to let it drain through, because it can't go right through the phospholipid bilayer because it's a charged electrolyte. So it has to be allowed to pass through a sodium channel. But when those channels open, sodium, all of this sodium that's been pumped out, wants to flood in. And when it does, it makes the inside positive again. This is what we call an action potential. An action potential is a reversal of the charges that of the resting. If resting is positive outside, action is positive inside. And it's temporary. And by temporary, I mean about 1/1000th of a second. It's about 1 millisecond that this lasts. 1/1000th of a second, this flip. And then it quickly, like 1/1000th of a second, and then for the rest of the second, it's back to being positive outside. But an action potential's real power is that it can cause a chain reaction. If I can get some sodium to come in here and make the inside positive, then I can get some other channels to open here, making the inside positive, which causes these to open here, making the inside positive, which can open these down here. So the real power of the action potential for communication is the chain reactions that it can cause. All of those chain reactions that it can initiate and cause wave-like down a membrane, across a membrane, or down along a membrane. Okay? So that's the real true power of these things called action potentials. Okay? All right. So this is, I'm going to have to erase it again because I've got too much mess up here. I don't mind just kind of rewriting it. So an action potential traveling along the neuron is actually a series of sodium coming in, which causes the inside to be temporarily positive, which causes sodium to come in, making the inside temporarily positive, which causes sodium to come in. Like one causes the next in a chain reaction. And they move down towards the axon terminal, When it gets to the terminal, I always look at this list and I'm like, man, it took me forever to explain 4.A, but I do want to make sure that you guys remember the difference between a resting potential and an action potential, because in chapter 12, we are going to talk about axons specifically again, and it'll pay off because we'll go over this stuff again, and I think it'll really start to sink in when we come back and look at it again. So thanks for your patience working through that. Here we go. Now this is where the real fun begins. We got an action potential traveling down this axon, and it gets to the axon terminal, which, as we said back on page 110, is really kind of the first part of this whole neuromuscular junction relationship. Okay? So the axon terminal's traveling along. Sodium in, sodium in, sodium in. At a certain point down here at the terminal, making the inside positive doesn't open sodium channels, it opens calcium channels. That's what these little spots represent. And calcium is also higher concentration outside, and if you open channels, it wants to diffuse in also. Okay? So up here on the neuron, we've got sodium diffusing in through sodium channels, which are not pictured on our diagram. But at the axon, what's unique about the axon terminal is it has these calcium channels, and calcium rushes in. Calcium causes these vesicles, which are represented by the circles on your diagram. These vesicles contain the neurotransmitter acetylcholine. Now, acetylcholine is just a chemical that's stored in these vesicles, but calcium causes these vesicles to do exocytosis, and the acetylcholine will diffuse across the synapse. And when it diffuses across the synapse, on the motor end plate over here are acetylcholine receptors. I'm sorry, I'm just going to have to represent it as an A. I didn't make my picture quite big enough. Acetylcholine will bind to these receptors. Those are called acetylcholine receptors. Okay? When the acetylcholine receptors have this is kind of like a lock and key. So acetylcholine is kind of like the key that clicks the lock, turns the lock, and it's attached to another channel over here. And this other channel over on the other side is a sodium channel. It's also a sodium channel. And so when acetylcholine binds, it lets some sodium in by diffusion, but now it's sodium coming into the muscle. Remember, this is the muscle over here. What we saw before was an action potential traveling down the neuron. And the bridge between the two, the chemical bridge between the two, was acetylcholine. Acetylcholine causes the sodium channels to open on the muscle side, letting sodium flood in. Now that makes the inside of the muscle cell more positive. And what do we call it when the inside of a cell becomes temporarily more positive? What is that called? Letter H, point 4H on page 111. When the inside of a cell becomes temporarily positive. Yeah, it's an action potential. It's an action potential. Yeah. It's the way a neuron's action potential was able to transfer an action potential over to this muscle cell. And just like it was a chain reaction coming down the neuron, it'll be the same thing in the muscle. So these sodium channels let sodium in, making it more positive inside, which will cause other sodium channels to let sodium in, causing this area to become positive, opening these sodium channels in this part of the membrane. They're all over the place, causing this to be more positive inside, causing this and this positive. And so you'll have this like wave-like movement of positives. Once it gets past this part over here, this part will go back to being negative inside again. Like I said, it happens just really quickly. So you have this wave-like positive inside, but what's different about a sarcolemma of a muscle is it's got these T tubules, these little manholes that go down inside, and that can carry the action potential down into the muscle, between and around the myofibrils, causing the inside to become more positive. Okay? When the inside becomes more positive over here, what we have over here is we've got sarcoplasmic reticulum. That doesn't show up very well, my bad. Let's see if yellow shows up better. No, not at all. Let's just make it dark. Sarcoplasmic reticulum that's loaded with calcium inside the cell. This is not the calcium from outside the neuron that we talked about before. This is calcium stored in the SR. And these channels right here, these are calcium channels that, again, when it gets positive around them, they will open. But this time, calcium floods out into the muscle fiber. So the action potential of sodium flooding in moves down the T tubule until it connects with these calcium channels on the SR. When it does that, it allows sarcoplasmic reticulum to flood all over the inside of the cell. And what do we need calcium to do? What have we needed calcium to do in this muscle that we talked about earlier before our break? Calcium is going to bind to what? What will calcium bind to? Binds to troponin, which stimulates troponin, because troponin is attached to what? It's going to bind calcium, and then when it binds calcium, troponin is going to pull tropomyosin out of the way. Yep, moves tropomyosin out of the way so that myosin can attach to actin, and this is where that calcium comes from. Okay, so before I switch to my next picture, which is the bottom picture on page 112, before I switch to that, let's take a second to kind of look at this diagram. This is what was needed to get calcium out there for troponin, to bind to troponin to get this whole thing going. This was the event that was needed. And what we really needed was we needed a signal from the neuron. And this back here was coming from the brain or spinal cord. If we follow this way back up here, we're going to find the beginning of that neuron somewhere in the brain or spinal cord that then decided it wanted this muscle fiber to contract. So it says, the brain or spinal cord says, I have control of that muscle, and if I want it to contract, I'm going to send a signal to it. And it does that by sending a wave-like action potential to the axon terminal. The action potential opens calcium channels, letting calcium in from the outside. Calcium stimulates exocytosis, release of acetylcholine, this chemical, across the synaptic cleft. Acetylcholine binds to the receptor, which, kind of like a key in a lock, clicks the sodium channels open that it's connected to. Almost like you put your key in your house door, you turn it, and it allows you to open that door, which allows you to go into your house. Acetylcholine is the key that turns the lock, which is the receptor, opens the door for sodium to come in, makes the inside of the cell more positive, and starts a wave-like action potential across the muscle fiber membrane, which then dips down into the T-tubules, which are attached to the calcium channels on the SR, opening those, letting the calcium that's stored at a high concentration go from high concentration out to low concentration, where it will attach to troponin, starting that whole cycle that we already talked about. Now, before I erase all of this, there's one other thing under point I on page 111. And that is, how do we stop this whole thing when we're ready for muscle to relax? We got to get acetylcholine out of the lock. We've got to get it out of here so that this channel will close, this sodium channel closes. To do that, there's an enzyme constantly floating around in the synaptic cleft. It is an enzyme actually made and secreted by the muscle itself, and it's floating around in here, and it's called cholinesterase. Its real full name, it's crazy. That's only its second name. Its full name is acetylcholinesterase, and it breaks down acetylcholine. It's almost like it breaks your key. It breaks the key. Now, I know that sucks. We've all done that, breaking the key in the lock. But this is a good thing, because it's going to take the key out of the lock and break it so it can't get in the lock, which will allow all of this over here to stop. It turns off these channels. Calcium gets pumped back into the SR. That's what's happening right here. These are called calcium pumps. This is calcium getting pumped back in for relaxation to get the muscle. It's taking the calcium away from troponin. Okay? Taking it away. So that allows the muscle to relax. That's a good thing, yes, which allows the muscle to rest, exactly. So we need that cholinesterase to actually break the key, to break the acetylcholine. Not to worry, because the key pieces, the acetyl group and the choline, the two molecules, they're going to get taken back up into the axon and reassembled. They're going to get put back together. So the calcium release from the SR, when the charge begins, positive inside, and then goes to the axon. No. These are different calciums. I want to make sure that I understand your question. These are not the same calcium at all. No. The calcium released from the SR is calcium that has been stored in the SR the whole time. The calcium coming into the neuron up here is just calcium floating out here in the space outside the cells, the interstitial fluid. This calcium coming in here is not the same. This is calcium that's stored here. Okay, yeah. Different. That's a good question. This is different calcium. All of your cells also have calcium pumps, and they try to always pump calcium either into the SR or out of the cell. Calcium has a tendency to want to crystallize and form calcium crystals of calcium phosphate, like we talked about in bone. before. And so most cells don't want a lot of calcium inside the cytoplasm. So it's just kind of a common thing for there to be a higher concentration of calcium outside of cells and trapped in sarcoplasmic reticulum. Okay? Now this is the bottom of that picture on 112. All right? And this is this top one here. I always giggle a little bit because I know this is Kathy Schultz's artwork. I'm pretty sure this is either Dr. Schultz or Ms. Long that drew this. So I think they did a great job. You can tell I like it because I took a picture of it and included it in my slides. I always like to teach directly from it. But I think it's cute. I think it's cool. But this up here is the muscle. This is the thin filament. Okay, so this is the Z line, thin, right here. So these little balls right here are actin, okay? And this is tropomyosin, and that little blocky thing right there is troponin, okay? But then you've got your picture up above where if calcium is coming out, if we got calcium coming out, it binds to the troponin. And look now, here's at rest, here's our tropomyosin blocking actin, and here's myosin sitting over here waiting, can't wait. It wants to magnetically connect to actin, but it can't. Tropomyosin's in the way. But here comes calcium. Calcium triggers troponin to move tropomyosin. That's now tropomyosin is out. See, look, it's out of the way. Up here, it was in the way. Now it's totally moved out of the way. And now the two magnets can get together. The magnets can stick to the fridge. And remember, that doesn't require any energy. They just stick magnetically. But they will start the cycle. Once they attach, then they will pivot, which causes them to release that ADP. And then ATP attaches to the head, causing it to reset at a higher spot, and it'll attach again, and then it pivots again, and every time it pivots, it draws the Z lines closer together. Every time those myosin heads pivot all along the thin filaments, it pulls the Z lines closer together, causing muscle contraction. Okay? And guys, this is what I said at the beginning of class tonight, this afternoon. All of what I'm talking about happens as fast as you can blink your eyes. Your eyelids are skeletal muscle. And yes, there is an involuntary part that your brain is in control of, but it's still voluntary skeletal muscle. And as fast as you can blink. Now, other muscles can't twitch quite as fast as your eyelids because those muscles are really small, and they have a special kind of fiber in them that are called fast twitch fibers. But I mean, as quickly as you can snap your fingers, those muscle contractions contract and then relax. And the whole cycle, the relaxation the contraction is stopped by that cholinesterase in the synaptic cleft. So all of this, page 111 across to page 112, is all about it's how fast all of this can happen. And it, like I said, it stuns me every time I think about it. Okay? Let's look at some of these questions, bottom page 111. I know your brain's fried, but we still got a little bit left. Whew. Dig deep, y'all. Dig deep. Deep breath. Where in the axon terminal is neurotransmitter stored? These are like ample short answer test questions. Where in the axon terminal is neurotransmitter stored? It's kind of on my picture. Vesicles. Yes, in those vesicles. Okay? What triggers its release? What triggers the release of the neurotransmitter? Calcium, calcium, calcium coming in. Calcium coming in triggers the release of calcium. How is the neurotransmitter released? How is the neurotransmitter released? Starts with an E. Exocytosis. Exocytosis. Right. So let me square this up. All right. The neurotransmitter is no. It's all right, Cheryl. It's all right, Cheryl. I'm glad you said that because you actually gave us the answer to the next question down past the one that says, does that require ATP? Okay, so let's back up a little bit. How is the neurotransmitter released? Exocytosis. Does that require ATP? Abso-freaking-lutely yes. You don't need the freaking part. Yes, it does. Oh yeah, exocytosis is a big energy demanding process. Yes, it does. However, next question. How does the neurotransmitter move? Everybody look at, I want Cheryl to get credit. Look up in the chat. She answered it ahead of us. How does the neurotransmitter move across the synaptic cleft, Cheryl? Diffusion. She was right. She was answering the wrong question. Diffusion. Okay? It's my fault for asking the wrong question, Cheryl. You got it. So, does that diffusion require any energy? Does the diffusion across the synaptic cleft? No, diffusion never requires energy. To what does the neurotransmitter bind? Acetylcholine receptors. How does that affect the charge across the motor end plate? I'd say causes an action potential. How does it affect the charge? I would write down causes an action potential. Because I want you to remember, now you can put in parentheses, makes it more positive inside. Makes it more positive inside the cell. You can put that. But I want you to remember that what we call more positive inside the cell is a positive inside. It's called an action potential. It's temporary, very briefly, yes, quick, milliseconds most of the time. Okay? It binds to the receptor, binds to the acetylcholine receptor. How is How is acetylcholine removed? And by the way, the answers to all of these are right above. I want to talk through them with you, but I want you guys to see the answers to all of these are right there above, except for the ones about does it require ATP or not. So how is acetylcholine removed from the synaptic cleft? It's right above, right there in bold. Cholinesterase. I'll probably call it acetylcholinesterase. That's the longer word is the one that I learned, but I'm trying to. What would happen if acetylcholine was released from the axon terminal, but could not bind to the receptor? What would happen if we got acetylcholine out of here, but it couldn't bind to the receptor? What would happen if acetylcholine did not bind to the receptor? There would be no muscle contraction. Big picture, yeah. No action potential on the motor end plate, no action potential across the sarcolemma, no action potential down the T-tubule, no calcium released from the sarcoplasmic reticulum, no calcium binding to troponin, no tropomyosin moved out of the way, muscle remains relaxed. Because the channels won't open. The question that's being asked there at the bottom of page 12 is, what happens if the key doesn't fit in the lock? Can you open the door? No. Sodium doesn't get in. So even if acetylcholine is released, you don't open the sodium channels. So nothing happens on the muscle side. You know what this is called? This is called myasthenia gravis. It's an autoimmune disease where antibodies from your immune system directly block the acetylcholine receptors. It's like if you came home and somebody had changed the locks on your door. You still have the key. We still got the key. We still got there's nothing wrong with acetylcholine. But somebody's blocked the lock. And what does that cause? No muscle contraction. Patients with myasthenia gravis typically present early in the process of this disease. Their early presenting symptoms are muscle weakness. Yeah, it's not body-wide paralysis at first. It typically shows up in the small muscles of facial expression. Droopy eyelids, actually feels effortful to smile. Eyelids are kind of droopy, or eyelids are difficult to open. That's how it usually shows up first, in the smaller muscles. And I mean, as soon as somebody notices those symptoms, they will often report it to the doctor. They'll go to the doctor, and those will be the presenting symptoms. Now, if they don't pay attention to it and allow it to progress, they'll notice body-wide muscle weakness and fatigue, what feels like muscle weakness and fatigue. Is it treatable? It sure is, yes. It is treatable, and often the treatment doesn't cure it. It often resolves itself, but the treatment can make it go away a lot faster. Yes. So what we have are antibody treatments that can block the blockers, if that makes any sense. You give them a drug that blocks these blue things that I've got drawn in blue. It blocks those from attaching to the acetylcholine receptors. Yep. I wanted you guys to know why we were asking that question at the bottom of 112. It is not a hypothetical question. Okay? What would happen, top of page 112? What would happen if acetylcholine could bind to the receptor, but could not be broken down by cholinesterase? Acetylcholine does bind, but it doesn't get broken down. What would happen? What do we need cholinesterase to do? Yeah, continuous contraction. Muscle just kind of, it contracts and stays contracted, no rest. Anybody familiar with this? You know what this is called? Constant muscle contraction. Can't relax it. It often, and again, like myasthenia gravis, often shows up early in smaller muscles of the face. In fact, I'm trying to give you kind of a clue. One of the early symptoms is inability to relax the jaw muscles. Inability to relax the jaw muscles. It's not Bell's palsy. Bell's palsy is inability to contract muscles. But this is inability to relax the jaw muscle. If you can't relax your jaw muscles, what can you not do? If you can't relax those muscles, what can you not do with your jaw? I mean, sure. Yeah. I'm sure. The muscle is contracted. It's contracted. You can't open your mouth. You cannot open your mouth. You've got bigger problems than you just can't chew and eat. Bam, Joshua got it. Yep, it's called lockjaw. Its real name is tetanus. It's caused by a bacterial toxin that interferes. It actually interferes up the line here in the central nervous system, but it keeps acetylcholine coming out and stops it from being broken down in the gap. And often the first symptom is this jaw muscle that is cramped and it doesn't feel good. It's muscle cramping, but you can't get them to relax. And those big muscles of your jaw, those are the most powerful muscles in your face. Ounce for ounce, the masseter and temporalis muscle are some of the strongest muscles in your whole body. Other muscles are stronger just because they're bigger. But if this masseter muscle on your jaw was the same size as your biceps brachii, it would actually be stronger than your biceps. If it was as big as your rectus femoris, of your quadratus femoris muscles, if it was as big as that muscle, it could move much more. So these are very powerful muscles. And when they tighten and lock, that can be very painful and very dangerous. So like I said, you've got bigger worries than just being, it's not that you won't be able to flap your jaw, you won't be able to even open your jaw with this. So again, I wanted to put it in the bigger picture of why we're asking these questions. We want you to understand and see why we want you to know these things. Okay. Other questions there, definitely important to know about. This is what the whole sequence of events looks like in your textbook. So if you've got the textbook, this is kind of the same thing, showing the same sequence. Right down here in the bottom right, they're showing us how this enzyme cholinesterase, you can see how they call it acetylcholinesterase, breaks down the acetylcholine to allow it to disappear. But this is all the same thing. It's just a little bit clearer set of pictures. So let's wrap it up with, I know we got a few minutes to go. I don't want to stop now, but we won't quite be finished with chapter 10 today. But there are some concepts I want to kind of rapid fire in the last few minutes of class, which is exhausting because it's like I've had you guys running a marathon today, and now I'm asking you to sprint for the last 15 minutes. So I don't even know if everybody's still awake. So hang in there. I'll tell you what. Let's do this. Brief break. I'm not going to walk away. Here's what I want to do. A friend of mine who teaches at Gwinnett Tech said that she likes coffee number 2. I'm past 2 coffees and on to a cup of tea as well. Mm-hmm. Yeah, Wanisha, you're right. It is a lot, but that's why I want to do it all as much as I can today so that you've got time to process it and study it. But here's what I want you to do. Everybody that's here today, I want you to emoji. This is what she did. Another teacher did just type here. This was the way she started taking attendance. This is how I'm going to make sure that you're still here and that you're paying attention, because I can copy the chat to check for attendance. I haven't done it before, but I'm going to do it from now on. Now you have to use a single emoji. Don't use multiple emojis to make dirty symbols. Okay? Use a single emoji from the set called Smileys and People. Use a single emoji from the set called Smileys and People. No fruits and vegetables. Use that in the chat to tell me how you're feeling right now. And everybody do it. Everybody. Here, I'm gonna put it up here. Everybody do it real quick. Oh, somebody did the ghost. That's cute. Oh man, that one looks constipated. Emoji your attendance. This will be a nice mental break before we do our last stretch run, right before the end. Everybody emoji your attendance today. That's how I know you're still here and paying attention and you're awake. Hello? Can you hear me? Some of you are I know some of you just log on and then you go off and turn on the TV over there somewhere. I know what y'all do. There we go. I like the alien. It's a good one. Oh, Yulieka, I like that I like the swirly eyeballs. Yes, that's good for confusion. That's a good one for confusion. I like the crazy looking robot. Good one. Krista's like, nothing. Krista's emoji would be this, dot, dot, dot. All right, have I reached everybody? All right, because I'm gonna keep a copy of this chat, and I'm gonna use this to count for attendance today. Okay, let's get back to the action. There you go, Lorraine. All right, coming in at the last second. Let's get back to where I left off, because I do want to talk about this bit at the top of page 113 and beyond, about what's referred to as a motor unit. A single motor unit is actually a bunch of cells working together, but it always involves a single neuron, one motor neuron, and however many muscle fibers it innervates. And here's the thing. We can have large, small motor units, medium motor units, because here's the thing. Take a look at this illustration from your textbook over here. So here's part of our central nervous system, our spinal cord, and there's some of those motor some of those motor neurons that are kind of painted different colors. We've got a purple one and a blue one and a red one. And let's see what the purple one does. It's going to send an action potential, sodium in, causing the inside to be positive, wave-like chain reaction along the axon, and then it splits when it gets into the muscle. And some of these axon terminals go to certain muscle fibers, but not all of them. Only the ones that are in purple. Now, your real muscles aren't really color-coded like this. This is just color coding so you can see the concept. This whole thing is a collection of muscle fibers. Now, I know your brains are fried, but earlier at the beginning of class today, what did we call, I'm going to try to ask my question really slowly to get you a chance to think about it, because I really do want you to think about it. What do you call a whole collection of muscle fibers? That's a fascicle. That's right. This is a fascicle. And what is this that surrounds a fascicle that I'm drawing? What surrounds a single fascicle? What is this called? Drop a clue right there. Perimysium. Okay, that's perimysium. Okay. Now, a single one of these purple things, purple, blue, reddish, okay, that's a single muscle fiber that is surrounded by endomysium. But right there is the neuromuscular junction for each one of these. That little spot, that's where we've been zoomed in all this time. Now we're zooming out a little bit to see that when, check me back up here on the spinal cord, when this cell sends an action potential, it will actually only activate 1, 2, 3, 4, 5, 6, 7 of these fibers in the fascicle, not all of them. Okay? When the blue one fires an action potential, it's only going to activate these 7 blue ones. And when the red one fires, it's only going to activate these 6 red ones. Okay? And this is how the spinal cord can control the fascicle. And I said this before, this is why each muscle fiber is surrounded by its own endomysium, so that maybe just the purple ones can contract without wrinkling or messing up the blue and the reds, so they can slide past each other. But this is how the spinal cord can get small contractions from a muscle, but can use the same muscle to get bigger contractions. Okay? So picture this. Picture this. I don't know if you guys can see me down here. Okay, I'm just using this because I got it handy. I got my cup of tea, and I can lift my cup of tea with my biceps brachii, okay? But I can also use that same biceps brachii just to lift my hand. Okay, so just to lift the weight of my forearm and hand, I can use biceps brachii, but I only use a few motor units to do that. To lift the T, I've got to lift a few more motor units. Okay, to lift my 4-year-old, I got to use a lot more motor units. To lift my 11-year-old, I gotta use them all, right? But that allows the spinal cord and the brain to use parts of muscles and then more if more is needed. Okay? So check it out. A few motor units at first, and then more if more is needed for the greater contraction, for the greater tone. Okay? That's what we call recruitment. And the spinal cord will recruit one motor unit at a time. So it might send an action potential just to the purple ones first, and if that doesn't make the object move, it'll send another one. It'll keep the purple one going and add the blue one, and then if the object doesn't move, it'll add the red one. Okay? And so that's called recruiting motor units. And again, what amazes me? Check this out. Watch this. Watch this in the camera. Watch. You see how fast that happened? I just lift that teacup right up. You see that? That's how fast recruitment happens. Not only does the muscle contraction happen fast, the spinal cord back and forth figures out how much it needs to move that object, and that recruitment happens quick. A large motor unit would be one motor neuron and a lot of muscle fibers, like 1,000 or more. 1,000 or more muscle fibers, all activated by one motor neuron. Okay? The large ones are the ones, it says, in 113, which muscles have large motor units with a large number of muscle cells per neuron, and why? These would be the muscles of your legs and hips, because you need large amounts of force to walk and run, and you need lots of muscle fibers at one time. So large motor units are for large muscles where you need large movements. Small motor units would be one motor neuron and 6 or 7 muscle fibers. Not even a dozen, just a very few. And these would be where you need fine, small motor movements. So where would these be? Well, your eyes, the movements of your eyes, and the movements of your fingers. Yeah, fingers. Dongsil knew where I was going. Yeah, the fine motor control of the muscles the muscles that move your fingers are mostly in your forearm, but those muscles use small motor fibers so that you get very, very fine control over a pen or a pencil and very, very tiny movements. Most of the others, like of the shoulders and the upper arms, the back and the torso, we'd call those medium motor units, and they can kind of do a little bit of everything. Okay, the all or none is there's no partial contraction, so that refers to these motor units too. If a single motor neuron sends an action potential to these muscle fibers will fully contract. But again, if it's just the purple ones, the whole muscle doesn't contract, just those 7 out of, 20 of them there? Just those 7 out of 20. So you won't get a full contraction of the whole muscle, but you will get a full contraction of those muscle fibers, of those individual muscle fibers. They will contract to their maximum ability. So dividing everything up into these motor units is a great way the body has of dividing the job, dividing the work. Okay? Muscle tone is how some motor units are kind of active at all times, but your spinal cord kind of switches some on and off. So some motor units are on when others are off, but this on and off even happens while we sleep. And the advantage of this is that it really kind of helps muscles to maintain a tone and ready to go when you call on them. So even if muscles are relaxed, you feel like they're relaxed, they're not actually 100% fully relaxed. If a muscle is completely under-relaxed, undertone, it's called hypotonic. And I know that sounds like a word that we talked about when we talked about solutions before, but it is a word that really actually means under strength, low strength, or under strength, because the word tone comes from a Greek word that means strength. So hypotonic is what we call a flaccid muscle, and this is usually damage to the, thank you, Jensen, damage to the motor neuron between the central nervous system and the muscle. Hypertonic is where it's over contracted, and that neuron can't stop the whole contraction. All right, so I got through as much of that as I possibly could. We're gonna the main topic that we'll have to save for next week is page 114, ATP needs in muscle contraction. Where does the ATP come from? Where does the energy come from? What happens with fatigue and the twitch, and the types of contraction. I'm going to cover all of that stuff relatively quickly at the beginning of class next week. And I want to tell you guys this before you go. I'm not actually going to go through in here. We're not going to have enough time to do cardiac muscle and smooth muscle on 118 and 119. So I definitely want you to spend time watching her videos on that stuff, because we're not going to be able to have time to and be able to move along. Nope, no problem. Thank you, guys. I'll stay and answer questions if anybody has. Thank you for staying overtime. I'm sorry to keep you guys late. Have a great weekend. Have a wonderful rest of your Friday, and I'll see you guys next Friday. Keep up the good work. Keep at it. Buckle down. Keep working hard, and I'll see you guys next week. Yes, thank you. Thank you. Have a great weekend. I'll hang around if anybody has questions. Take care. Thank you, guys. Thank you all for coming. Thank you so much.